Prepare to rewrite your understanding of human history. Genetic evidence reveals a startling truth. Every single person alive today descends from a single woman. A woman who lived in Africa tens of thousands of years ago. Discover the incredible story of mitochondrial Eve and how her legacy connects us all. Across multiple religions and cultures across the world, it is believed that we all come from the same parents: Adam and Eve, Manu and Shatarupa, Ask and Embla, Fushi and Nuwa, or Deucalion and Pyrrha. It is clear that although accounts may differ from across the globe, there is a strong belief that we all come from the same parents. A single thread connects us all, and we all come from the same source. While there is no definitive research on who the Adam and Eve of the human race were, we do have the next best thing: mitochondrial Eve. Although not our first parent, mitochondrial Eve is the most recent common matrilineal ancestor of all living humans. Identified through the study of mitochondrial DNA, or mtDNA, she is our universal grandmother, regardless of race, ethnicity, religion, or nationality. She is essentially the first of a long line of unbroken maternal ancestry that leads to all current humans, including you and me. Estimated to have lived between 150,000 and 200,000 years ago in Africa, it is essential to clarify that mitochondrial Eve was not the first human, nor a singular mother in a strict biological sense. Instead, she represents just one part of a huge family tree where many women and groups of people lived at the same time as her, all contributing to the genetic makeup of modern humans. But how exactly did we trace every human back to her? Well, to do that, we had to do some really complex science that, honestly, sounds like magic. For anyone who took a biology class, the words "the mitochondria are the powerhouse of the cell" are probably etched somewhere at the back of your mind. But the mitochondria are honestly so much more. For one, the mitochondria have their own DNA separate from your normal DNA. Now, this sounds weird, and anyone with biology knowledge is already asking, isn't DNA the building block of life? How do the mitochondria have their own DNA? The answer is as simple as it is confusing. In a weird, unexpected twist, the mitochondria weren't always part of our cells. They're believed to have originated from ancient bacteria that were engulfed by a larger cell in a process called endosymbiosis. Over time, these bacteria became a permanent part of the cell, eventually evolving into the mitochondria of today. So how does this explain the DNA? Well, because of their bacterial origins, mitochondria have their own DNA separate from the cell's nuclear DNA. This mitochondrial DNA is what we need to focus on today, as it was the key to mitochondrial Eve. See, each mitochondrion contains multiple copies of mitochondrial DNA, or mtDNA, which is much smaller than your usual nuclear DNA. This mtDNA has a weird hidden feature. It can only be inherited from the mother because sperm lose their mitochondria during fertilization, meaning that all mitochondria in a child come from the mother's egg. Besides the fact that this means you share the same mitochondrial DNA with the woman in your lineage, this property is super important to science as it makes mtDNA a direct genetic link to one's maternal ancestors, essentially allowing studies in lineage and ancestry. How? Well, because mtDNA is passed down generation after generation through maternal lines. Analyzing mtDNA enables scientists to trace back through familial lines and determine common ancestry. This property is beyond crucial to fields like evolutionary biology, anthropology, genetics, and, of course, mitochondrial Eve. See, the concept of mitochondrial Eve relies almost completely on mtDNA analysis to find the most recent common female ancestor of all currently living humans. By studying variations in mtDNA, scientists can find out just how different populations have emerged and evolved over time. But why not use conventional DNA? Conventional nuclear DNA isn't used for this study because, in contrast to mitochondrial DNA, nuclear DNA is inherited from both parents. This means it comprises a mixture of genetic material from the mother and the father. Every individual receives one set of nuclear chromosomes from each parent, resulting in a combination of traits. This biparental inheritance means that nuclear DNA reflects the contribution of both maternal and paternal lines. Although it is great for reproduction and studies of that kind, it is terrible for the study needed for mitochondria Eve, as this DNA changes in each individual and it creates a complex genetic profile for each individual. Essentially, twins can have different nuclear DNA but must have the same mtDNA, making mtDNA more suitable for this kind of maternal study. All good? Great, so now we have to ask though, how did we even find mitochondrial Eve in the first place? After all, science is built on the back of discovery. Now when you hear mitochondrial Eve, it's easy to think she was the last woman alive after an extinction event, and we're all offsprings from her. But that isn't the case. During her time, she was just one of the many women who called Africa home. She, however, is the last common ancestor in an unbroken matrilineal line from whom all living humans today have inherited their mtDNA. This doesn't mean that she was the only woman alive during her time. In reality, many other women existed, but their lineages did not persist in the same manner as hers. Consequently, mitochondrial Eve represents a genealogical endpoint rather than a first woman. But how did we find her? As we've said already, the identification of mitochondrial Eve involved the analysis of mitochondrial DNA. To trace human ancestry, researchers use a method called the molecular clock. Interesting name aside, this method is based on the fact that mutations in mtDNA happen at a fairly steady rate over time. And so by comparing the differences in mtDNA between various people, scientists could estimate how long ago their common ancestors lived. Using this approach, they built phylogenetic trees, which are diagrams that show how different groups of people are related and how their lineages split from one another over time. These sophisticated versions of grade school family trees helped scientists map out the human family tree and understand how various human populations are connected. Looking at the results, mitochondrial Eve was determined to be the most recent woman from whom current humans can trace their maternal lineages without interruption, indicating that all living humans share an unbroken maternal ancestry leading back to her. Among the studies that looked into mitochondrial Eve, one of the most prominent was the study connected by Rebecca Kahn, Mark Stoneking, and Alan Wilson in 1987. This particular study was monumental in popularizing the concept of mitochondrial Eve, as the researchers published their groundbreaking paper titled Mitochondrial DNA and Human Evolution in the journal Nature. This study involved analyzing mtDNA from 147 individuals across 5 geographic regions, including Africa, Asia, Australia, Europe, and New Guinea. By comparing the mtDNA, they demonstrated that all current human mitochondrial lineages could be traced back to a common ancestor residing in Africa approximately 200,000 years ago. Outside the common grandma theory, their research was pivotal in supporting the theory of recent human origins out of Africa while establishing a scientific framework for understanding human evolution. The data they collected not only indicated a common ancestor, but even suggested that the majority of mtDNA diversity is found within African populations. Essentially, it proved that Africa was ground zero for who we are and our evolutionary history. It was the wide-reaching implications of their study that captured public attention and ultimately attached the label Mitochondrial Eve to our shared mother. You and every individual are the offspring of a man and woman, and as such, if we have a mitochondrial Eve, shouldn't we have a common Adam? Quick pause. If you're enjoying this journey through the prehistoric world, don't forget to like and subscribe. More than 97% of our viewers watch without subscribing, and we'd love to have you join our tribe. It would make all the difference. So, is it done? Great. Thanks a ton. Much like mitochondrial Eve, Y-Chromosomal Adam is recognized as the most recent common paternal ancestor of modern humans. And like Eve, he was traced exclusively through not mtDNA this time, because that would be impossible, but the Y chromosome, which is passed from father to son. This means that only males inherit this genetic material, creating a specific lineage that tracks male ancestry. Similar to mitochondrial Eve, Y-chromosomal Adam represents a point of lineage convergence rather than a singular individual. He is estimated to have lived between 200,000 and 300,000 years ago. Looking at the timelines, it might seem like mitochondrial Eve and Y-chromosomal Adam could have lived at the same time or even been together. However, that wasn't the case. They each lived in different periods of human history, separated by quite possibly thousands of years. Their separate timelines just show the different genetic paths humans took as they evolved and adapted over time. While estimates suggest that Y-chromosomal Adam's life may overlap with mitochondrial Eve's, they did not live at the same moment. Their lineages are just the most recent common ancestors for the paternal and maternal sides of our genetic history. So what makes Eve so important? What can they tell us about ourselves? Today, mitochondrial Eve is crucial for understanding human evolution, and that's because of her connection to our evolutionary tree. See, the connection of mitochondrial Eve to the evolutionary tree is evident in how mtDNA varies among different populations. The analysis of mitochondrial genetic material allows researchers to pinpoint the approximate time frame of her existence, and even suggests that her haplogroup, basically a set of mtDNA sequence variations that have evolved over time, gave rise to the diverse mitochondrial lineages present in modern humans today. In layman's terms, she supports the out of Africa model of human migration. But that's not all, because since mtDNA accumulates mutations at a predictable rate, researchers can estimate how long ago different populations diverged from one another by measuring genetic differences. This essentially allows us to track it all back and look at the routes human populations took as they spread across different continents. This study, in turn, aids in tracing various ancestral patterns and connections among diverse groups. She essentially gave us this new ability to connect genealogical patterns using mtDNA, ultimately transforming our approach in ancestry research, enhancing not only our understanding of individual family histories, but also composing a broader narrative of human migration and interaction. But what do you think? Does mitochondrial Eve truly reveal our shared human ancestry, or is her story just one fascinating thread in the vast tapestry of evolution? Let us know your thoughts in the comments below. And while you're here, don't forget to hit that like button and subscribe for more deep dives into the science that shapes our understanding of the world. And if you're curious about how the out of Africa theory fits into this evolutionary puzzle, click on this video right here. Until next time, stay curious.